Hi everyone, welcome to BioBot classes. In our last video, we learned about mitosis, how our body cells divide to help us grow and repair. But what about the cells needed for reproduction? That's where meiosis comes in. Meiosis is a special type of cell division. It happens only in cells that make gametes like sperm in males and eggs in females. Now, here's the key part. Unlike mitosis, where the number of chromosomes stays the same, meiosis cuts the number of chromosomes in half. Why? So that when the sperm and egg come together, the new life starts with the correct number of chromosomes. Also, meiosis creates variety. That's why no two siblings look exactly the same. In this video, we will explore how meiosis works and why it is so important. Let's begin. Meiosis happens in two main stages, meiosis 1 and meiosis 2. You can think of it as two rounds of division, but only one round of DNA copying. You can think of it like this. The cell divides once to reduce the number of chromosomes and then again to separate the chromatids so that in the end we get 4 unique cells. Let's look at meiosis first. This is called the reduction division because this is where the number of chromosomes is cut in half. First comes prophase 1. It is important and longest phase of meiosis. It is where the real magic of genetic variation happens. Meiosis 1 is divided into 5 substages. First is lactotin. First, the chromosomes become visible under the microscope. Each chromosome has already copied itself. Now, it looks like an X with two sister chromatids. Second stage is zygoting. Something special happens here. Homologous chromosome, one from mother and other from the father, come together in a process called synapses. A structure called the synaptonemal complex is formed between them. The complex formed by a pair of synapsed homologous chromosomes is known as bivalent because there are two homologous chromosomes, or a tetrad because there are 4 sister chromatids. Next is packaging. While they are paired, they actually exchange small pieces of DNA, and this process is known as crossing over. This leads to genetic recombination. It creates new combinations of genes. This is why no two siblings are exactly the same. As you can see here, pieces of DNA has been exchanged between these two chromosomes. Moving to next stage, which is diplotin. In diplotin, the synaptonemal complex dissolves. Homologous chromosomes start to separate, but they stay attached at chiasmata. What is chiasmata? It is the point where crossing over occurred in the packaging stage. Moving to next stage, which is dikinesis. In dikinesis, chromosomes are fully condensed and they separate at chiasmata sites as well. Nuclear membrane breaks down and the spindle apparatus forms. Now let's move on to metaphase 1, another exciting step in meiosis. In this stage, the homologous chromosome pairs, the one you saw in prophase 1, line up in the center of the cell. Next, spindle fiber from opposite sides of the cell attach to the centromeres of each chromosome. This is getting the chromosome ready to be pulled apart. Next comes the anaphase 1. This is the moment where the actual separation begins. The homologous chromosomes separate. Remember, one from the mother and one from father are pulled to opposite sides of the cell by the spindle fibers. But notice the sister chromatids stay attached. We are not splitting the X-shaped chromosomes yet. We are just separating the pairs. This is super important because now each side of the cell will have half the number of chromosomes, and that's how meiosis reduces the chromosome number. So in short, in anaphase 1, chromosome pairs separate, but chromatids stay together. Now we reach telophase 1. At this point, the homologous chromosomes have moved to opposite poles of the cell. Next, a new nuclear membrane may form around each group of chromosomes. The cell then begins to split into a process called cytokinesis. By the end of telophase 1, we have 2 haploid cells, each with half the original number of chromosomes. Remember, the sister chromatids are still attached. They will be separated in the next round of division, which is meiosis 2. After telophase 1, the 2 haploid cells enter a short resting stage called the interkinesis. This is just a pause between meiosis 1 and meiosis 2. Here is the key point. During interkinesis, the DNA does not replicate again. The chromosomes are already duplicated. The cell is simply preparing for the next division, where the sister chromatids will finally be pulled apart in meiosis 2. Now, let's begin meiosis 2, which is very similar to mitosis. We start with prophase 2. At this point, we already have 2 haploid cells from meiosis 1. Now, in each of these cells, the chromosome, which still have 2 sister chromatids, start to condense again. The nuclear membrane, if it had reformed during telophase 1, now breaks down once more. Spindle fibers begin to form again, getting ready to pull the sister chromatids apart in the next stages. So basically, prophase 2 is same as prophase of mitosis, but now it happens in 2 haploid cells. Next is metaphase 2. Now, in each of two haploid cells, the chromosomes, still with their two sister chromatids, line up at the center of the cell along the metaphase plate. This is very similar to what we saw in mitosis. The spindle fiber from the opposite poles attached to the centromeres of each chromosome, getting ready to pull the sister chromatids apart. Now comes the anaphase 2, a very important stage. Here the sister chromatids, which were still attached, are finally pulled apart by the spindle fibers. One chromatid from each chromosome moves toward one pole and the other moves toward the opposite pole. This is just like anaphase of mitosis, but it is happening in haploid cells. By the end of anaphase 2, each pole of the cell will have a complete set of single chromosome. Finally, we reach telophase 2, the last stage of meiosis. Now, at each pole of the two cells, the chromosome begin to decondense. A new nuclear membrane form around each set of chromosomes. Cytokinesis occurs, meaning the two cells split again. And what do we have at the end? Four haploid daughter cells, each with half the original number of chromosomes. Each of these four cells is genetically unique thanks to crossing over, and that completes the amazing process of meiosis. Now, let's quickly understand why is meiosis so important. First, meiosis reduces the number of chromosomes by half. This is very important because when the sperm and egg come together during fertilization, the new organism will have the correct number of chromosomes. Second, meiosis creates genetic variation. How? By crossing over during prophase 1, where the chromosome exchange segment of DNA. This ensures that each gamete and each new individual is genetically unique. Finally, the meiosis is essential for sexual reproduction in all sexually reproducing organisms, whether it is plants, animals, or humans. Without meiosis, there would be no variation and evolution would not be possible. And that brings us to the end of the session on meiosis. Remember, this is a very important topic both for our understanding of biology and for our exams. If you found this video helpful, do give it a like and share it with your friends. If you haven't yet, make sure to subscribe to Bio Board Classes for more such easy explanation. I'll see you in the next video. Until then, keep learning and stay curious.